Meiosis is the subject of our lecture here, and while it's quite similar to mitosis, there are just a few respects which make it unique. To begin, meiosis is the cell process that produces gametes, and gametes are the sex cells, so this would be the sperm and the egg. Not to be confused with somatic cells, those would be all the other cells in your body. So the vast majority of cells in your body are made by mitosis. They are considered somatic cells, but in a small region in your body there are gametes that are also made. Those are very important, even though there's not as many of them, and they're made through meiosis. Okay, this occurs in the gonads, so the only place in your body where meiosis should be occurring is in the testes in the male, and then in the ovary in the female. And the thing that makes this process unique is that it converts a diploid cell into a haploid cell. If it was mitosis, it would just be a copy of the diploid cell into another diploid cell. Okay, how this occurs varies depending on if we're talking about animals or plants or fungi or what have you. For example, plants have a little bit of an interesting life cycle where they can actually have haploid cells called spores undergo mitosis to create an organism that's haploid, and then that organism that's haploid can then produce the gametes, which come together and form a diploid structure. So you actually have different structures in plants composed of either all haploid cells or diploid cells. That's not how it works in most animals, and certainly not in humans. In humans, we only just make the sperm and the egg cells. They never come together and undergo mitosis to make structures in our body that are haploid. But in plants, this can occur. An interesting example of this would be moss. So if you've ever been out in the woods in the springtime and you see some moss, the mossy leafy stuff is the structure that's made up of haploid cells, and then occasionally there will be a structure that comes out and is made up of diploid cells. So here we've got different structures here that have either haploid cells or diploid cells in them. So all that to say, it can vary quite a bit, but in humans, pretty straightforward. Just basically meiosis is used to make gametes. The gametes never come together to make haploid structures. They come together to make a diploid structure. Okay, there are two divisions in this process, which is one of the things that makes it unique from mitosis, and it is the fact that there are two divisions, which is why this process yields haploid cells rather than diploid cells. Here are the processes here. So there's basically meiosis 1 and then followed by meiosis 2. It's going to look exactly like mitosis did, where you have prophase, metaphase, anaphase, telophase, and there should also be a prometaphase, just like there is in mitosis, but it's not included in here. But it's not really all that important to pay attention to at this point. Okay. Now, after the first division, there will be one cell which is then going to divide into two new cells. That would be what happens in meiosis 1. And in the cell that starts is diploid, and then the cells that end up here are haploid. Again, diploid and haploid, the difference between the two, just something that diploid, as labeled here 2N, would have two sets of chromosomes, one from mom and one from dad, and then a daughter cell that's made here would just have one set of chromosomes if it was haploid. Okay, then in turn, each of these daughter cells will undergo a division to create two new cells, which are also haploid. So altogether, this process yields 4 cells. Now and they're all haploid. Start with a diploid cell, you end up, and they're all haploid. Now, another interesting feature here about meiosis is that all of these daughter cells are unique. They do not they are not genetically identical with one another. That is not the case in mitosis. In mitosis, the daughter cells are all going to be genetically identical with the parent. Here, they're all unique, and that's important because it allows for different types of gametes to be made. For example, if your mom and your dad all had the same genetically identical gametes, then anytime a sperm and an egg came together, it would create an individual with the exact same genome. And so if you have a sibling, the reason why you and your sibling are different is because of the vast array of different types of gametes that your mom had and vast amount of gametes that your dad had. Okay, we'll talk a little bit more about how that genetic diversity is introduced here in just a minute. All right, so meiosis prophase 1 looks something like this. Here we get the same story as mitosis. The nuclear envelope is eroding and the chromosomes have condensed. The chromosomes have already undergone S phase, so they are duplicated. So you'll see there's two sister chromatids where there used to be just one chromosome. So that's how we know that it's completed the S phase. Now, one of the unique things that happens in meiosis is that there's this process known as synapsis, which occurs. And synapsis is a process where homologous pairs of chromosomes come together to form what is known as a tetrad. And that's what we're seeing in this picture. Remember, homologous pairs of chromosomes are chromosomes, the one that you got from your mom and the one that you got from your dad, that are about the same size and bear the same bear information coding for the same traits. So because these are each duplicated chromosomes, that means there's technically 2 chromosomes for each on each side. That means that there's 4 altogether, which is why this is called a tetrad. Tetra meaning 4. All right, so this is all going to happen in prophase, and then another really important thing happens. The sister chromatids can actually swap DNA between homologous chromosomes. So we're seeing that somewhat here, where we've got this little blue chunk here starting to attach to this chromosome and this red chunk starting to attach to this blue chromosome. That's called crossing over. And this is basically reason number 1 that all the gametes are genetically unique, because crossing over occurs somewhat at random. And so because there's this random mixing that occurs between homologous pairs of chromosomes, which eventually will get separated out, it ensures that all the gametes don't receive the exact same information. There's a little bit of a variability there that can get packaged up into the cells. Okay, so reason number 1 why you and your sibling, if you have one, are unique. Okay, in metaphase then, this looks pretty similar to mitosis. You can see we have completed the crossing over here, so we've got some chromosomes mixing and matching. Now, one of the things that is going to make this unique from mitosis, so again another star here because this is a unique feature, is that the homologous chromosomes line up side by side. So that doesn't happen in mitosis. If this was mitosis, all of the homologous chromosomes would be in a single file. But that's not the case here. They're lined up side by side. And it's that very feature which is why we go from a diploid cell to a haploid cell in meiosis 1. Because of the fact that we have one set of homologous chromosomes here, and we have another set here, and those 2 sets are going to get separated. So this is the event that brings us from a diploid cell to a haploid, to pulling each set to a different side of the cell so it can become haploid. Note here on the figure that sometimes the red chromosome is on this side, other times the blue chromosome is on this side. this is another important reason why you and your sibling, if you have one, are genetically unique. This is called independent assortment. And independent assortment, and reason number 2 for the unique features of gametes. Independent assortment just basically means when these homologous pairs line up, it's random whether this one gets on this side or this one gets on this side. It either can occur, and that will make a big difference on all of the genetic information that's brought to this side of the cell versus this side of the cell. So whether a cell is going to receive one trait from mom or one trait from dad really depends on which of these homologous chromosomes it inherits. And that depends on which side the chromosome is facing when they line up at the metaphase plate. Now, it doesn't mean that one cell will get some information and the other cell will not. In theory, each cell should get a complete set of chromosomes. It's just that on that set of chromosomes that it received, there will be a mixing and matching of which percentage of the genes came from mom's original set and which percentage of the genes came from dad's original set of the chromosomes. Throughout this whole figure, you can imagine, if it helps, you can imagine that any chromosome that's blue is a chromosome that originally came from dad, and any chromosome that's red is the one that came from mom. So this is a diploid cell. It could represent any cell in your body, but specifically it's showing a cell that is in your gonads undergoing meiosis, and that cell would have still in it a copy of your dad's genome and your mom's genome all the way back from that first fertilization event. And then those sets will get separated. Okay, that's metaphase 1. Here's anaphase 1. We're seeing the homologous chromosomes now being separated. And now we have 2 haploid cells, because you can see that this chromosome is the long one, and there is no second, even though it's duplicated still, there is no second chromosome to match it. Same for the short one and same for the middle-sized one. And we can also see that they've exchanged some chunks of the DNA. So this is a result of crossing over. And then the nuclear envelope is going to reform to bring us to the conclusion of telophase 1. Okay, now we have 2 cells, and right after the nuclear envelope forms, then it's getting ready to divide again, so it disappears. And meiosis 2 is pretty much identical to the process of mitosis. So the thing that makes meiosis unique is basically meiosis 1, specifically all the stuff that's happening in prophase and metaphase, the synapses and the tetrads, the crossing over, and the way that they line up in the middle of the cell during metaphase. Okay, so just if we play this all out, here this looks again very similar to mitosis. We have duplicated chromosomes lining up. The big difference here would be that the sister chromatids are not genetically identical like they are in mitosis because of that crossing over that did occur. Okay, and then basically just pulling the sister chromatids apart, and that will yield 4 cells, which are all haploid and have a single individual chromosome. No longer are they duplicated. Okay, so that is the process of meiosis. Okay, that is the process of meiosis. Sometimes it's helpful to see this in a 3D animation, so if you think that would be helpful for you, I'm sure there's a whole bunch of them online you could find to see it happening sort of in real time, so to speak. Okay, a little bit more here about where this is occurring and how it occurs. Spermatogenesis, we said, occurs in the gonad, the testy, which is pictured here. And basically what's happening is in the testy there is this vast array of tubing, and those tubes are called the seminiferous tubules. And those are actually the site of sperm production. So if you were to take a cross-section of one of these tubes and look at it, it would look something like this. So this is the outside of the seminiferous tubule. And very interesting how it works. There are a bunch of cells that are on the periphery here. So each one of these little things here is, each thing with a dark circle is a nucleus, so that's a cell. All of these cells are diploid, and these are specifically called stem cells because they can undergo meiosis, and they supply the cells required to continue to make gametes. So how it works is that these will be undergoing mitosis to copy themselves. So imagine this is a diploid stem cell, and it's just gone through mitosis to copy itself. So now there's two copies of it. One of those cells will stick around to replenish the next generation. The other one then will undergo meiosis. And what's happening is, as the cells migrate closer to the middle of that tube, the seminiferous tubule, they're undergoing the process of meiosis. And this is the reason why some of these nuclei look a little bit, or at least the cells look a little bit smaller than they did way back here, because those are cells that have just recently divided, so now they're like half the size of the original parent. And you might ask, well, how is it that these cells know to undergo meiosis and not mitosis? It's a really good question. I don't know exactly all of the factors. I'm sure it's extraordinarily complex, but what I do know is that there is a whole bunch of chemicals and different types of things released here which will program the cell to undergo meiosis instead of going through mitosis, which would be the normal way that a cell would want to divide, basically. And it's very important that meiosis only occurs here. If meiosis happened in other parts of your body, it would create cells that are haploid in places that they shouldn't be, and it would just cause a whole lot of chaos in your body and in the development and function of your organs. So it's really important that meiosis only happens here in these tubes and nowhere else in the body. So there's actually a pretty good testy blood barrier here, that all of the chemicals and things that are released here don't diffuse into the blood and make their way into cells throughout the body. Otherwise, those chemicals could start programming other cells to undergo meiosis as well. So everything's kept pretty well under wraps here in the seminiferous tubules. Okay, in the female, this happens in the ovaries, and it's a little bit different because there's basically the precursor to the gamete, and what happens is every month a few of these, whatever they're known as primordial follicles or a primary follicle, they'll be selected to undergo what's called follicular development. And it's similar. It's just that whereas here you have the cells undergoing through meiosis, and then there's 4 complete cells produced. I should back up to what you're seeing here. These are little sperm that are mature, and so basically as the cell gets to the inside lining, it's now the haploid gamete. It's released, and then it later on becomes a fully functional sperm cell. Okay, here that precursor cell that could turn into the gamete is a little bit different because it's surrounded by this this border of cells. Here, the gamete is just made and then released. Here, the gamete is actually surrounded with a shield of cells, and those cells will be releasing different hormones that will program this, the precursor cell, to be undergoing meiosis. And when that precursor cell is eventually going to leave the ovary, which is known as ovulation, it still has some of those cells around it as a barrier. So you can see a little bit here of the histology on a slide. We've got the primordial follicles here, and there's another one here, and then you can see that layer of cells. And in here is an ocean of different chemicals, so to speak. It's a pool of different chemicals which are all working to cause this cell to undergo meiosis and not mitosis. Again, another isolated part of the body here so that these compounds don't make their way into the bloodstream. All right, spermatogenesis and oogenesis will have a couple of big differences. So spermatogenesis, the process of making sperm in the male, this will start when the male is about puberty. And as soon as it starts, the male will just continue to make gametes to the tune of millions and millions every single day until the day he dies. Oogenesis is a little bit of a different story. So by birth, a woman has all stem cells they'll have. Okay, so there's a set amount of how many eggs can be produced, because by the time a woman is, by the birth of that individual, there's a set bank of how many of these cells are available to then undergo meiosis. In the male, we always had the stem cell dividing, and then one of them would replenish the stock and the other one would then undergo through meiosis. In the female, the stem cells all divided and then went through mitosis and replicated and duplicated to a big, large supply of stem cells, and then from there, every single stem cell will be committed to undergoing meiosis. If it does, there isn't that replenishing that happens here in the male. So by birth, a female will have all of the stem cells ready to then go through meiosis, and they actually, in fact, do begin meiosis, and then they're all stopped. And so by birth, there's about 500,000 of these cells that are arrested in meiosis 1, and so that's in each ovary. So that's about 1 million of these cells which can undergo meiosis. It's a finite amount, and that number will decrease and decrease and decrease by the time a woman is of age to be in puberty. But even still, by about puberty, I think this number is something more like 200,000 in each ovary. So there's still quite a few cells in the ovary that can then carry onward through the process of meiosis. But they're all stuck in meiosis 1 here. That doesn't happen in the male. In the male, if a cell is going through meiosis, it just goes through it. It doesn't get stuck. Here, they're all stuck at meiosis 1, and only a couple of months will then be selected to start continuing on the process of meiosis, and that doesn't even really happen until puberty is reached. So those are the big differences between how gametes are made in male or female anatomy. All right, so because there is this somewhat of a shelf life here of gametes in the female, and it sort of erodes as a woman ages, there is a bit of a time span where fertility is optimal, and then it starts to wane once a certain threshold is passed. There's an interesting graph here which basically shows the fertility rate of a couple and the rate of spontaneous abortions. So that would be like a miscarriage. And you can see right at about 35 is where the inflection point is of the curve. Once we get past about 35 into 39 years old, the number of pregnancies that are occurring is decreasing, and it's becoming more and more likely that those pregnancies would lead into a miscarriage. And for whatever reason, it shows that one of the things that I was reading was that since the 80s, the birth rate in women older than age 35 has increased by nearly 60%. So on average, it appears that women are having kids later in life. That's something that I would say is true just of my own life, knowing that my friends and such. My wife and I, we had kids, started having kids when she was in her late 20s and I was 30, and we were like the only ones. So we were young ones then, and I don't think that was necessarily the commonplace for our people that were having kids in the 80s and the 90s. So I'm not really sure what the shift is there, but so part of this reason is for, again, the fact that those precursors to forming eggs are being depleted, and they're becoming aged. They're not continually replenished and renewed. Now, that doesn't mean that the age of a man does also not affect fertility. So when people study fertility, they might consider things like sperm count in the male. And sperm count is important because it requires actually a lot of sperm to fertilize an egg. Here we're seeing a picture of an egg, and there is this barrier around the egg, and the sperm have to collectively work to break that barrier. Sperm have a little capsule at the front of their head that has enzymes in it, and those enzymes will break collectively the armor off of the eggs, and until eventually one sperm can penetrate in, and then it shuts all the other sperm out, and now we have a fertilized zygote. So there are a lot of sperm that have to make the egg, and that battle is a losing battle for sperm. Sperm, the pathway from the pathway to fertilize an egg for a sperm is a very perilous, dangerous journey. And while millions and millions and millions of sperm might start that journey, most of them will be killed, and leaving just a select few. And so, because of that, if there's not enough sperm at the beginning, it could be the case that too many of them die in the process, leaving not enough to complete the race. So it is kind of a probability thing that men with a higher sperm count would have a greater chance of fertilizing an egg, and that chance declines as a man ages. Some of the reasons here I came across in a study were talking about that they have different acquired medical conditions, or there's a decrease in the semen quality, or increasing rates of DNA fragmentation that are all seen to increase in the male reproductive anatomy as a man ages. But I don't think that those changes are as significant as they are in the female anatomy, just for the sake that men are continually replenishing the supply of gametes that they're producing. All right, we'll finish up here just talking about various abnormalities that can occur in meiosis, the biggest one being non-disjunction. So what non-disjunction is when a chromosome doesn't find its way to the proper cell. So consider this: we have a cell, and here are the two homologous chromosomes lined up on either side. What should happen is that each homologous chromosome should make its way to a new part of the cell. So this one should go this way, this one should go this way, and this one should go this way, and that one go this way. In this case, one of them was pulled wrongly to the other side, and that can have really grave consequences later on the line. So that's the definition of non-disjunction, when they don't separate out as they should, and one gets carried incorrectly to the other side. So if we were to see how this played out, what it would lead to is all of the cells having an inappropriate number of chromosomes. One would have too many, or sorry, two cells would have too many chromosomes, and two cells would have too few chromosomes. Now, the severity of this event varies depending on where it occurs. If it occurs in meiosis 1, it's the worst, because we can see all of these cells end up with an incorrect number of chromosomes. If it happens later on in meiosis 2, so for example, maybe meiosis 1 is okay, and then during anaphase of meiosis 2 is when the non-disjunction occurs. Basically, the sister chromatid should have ended up in this cell, but instead it ended up in this cell. Well, if that was the case, then these would be okay, and then it's just that this other cell would have one too many and one too few in those daughter cells. Now, generally, this would be sort of a cause for a spontaneous abortion. The cell would not be able to survive, or the gamete would not be able to survive. But there are cases where specifically on chromosomes that are small and don't contain a lot of genetic material, or the sex chromosomes, where this a gamete in which this has occurred can survive and even go on to be fertilized by a sperm, and vice versa. Okay, a couple of examples here would be monosomy or monosomy, depending on how you want to pronounce it. One example of this is Turner syndrome. Turner syndrome is an individual that has one X chromosome and no other sex chromosome. So generally, In the female anatomy, there'd be two X chromosomes, and in the male anatomy, there'd be an X chromosome and a Y chromosome. And because the X chromosome doesn't contain as many, I guess one could say, essential genes, and that there is sort of a redundancy in having two of them in the female anatomy. If a woman did just only have one X chromosome, it would result in Turner syndrome, so that individual would still be able to survive, and there would just be a couple of consequences of not having the second X. So some of the female reproductive hormones would be in more short supply, basically. Okay, in trisomy, this is instead of having one too few, this is having one too many of a specific chromosome. One example is Klinefelter syndrome. This would be where an individual had an X and an X and a Y. So they had X and Y. The Y would render it male anatomy, but because there's another X chromosomes there, it's male anatomy with some excessive female hormones being produced. Another example is Down syndrome, which is three 21st chromosomes, so it's sometimes called trisomy 21. And if we look at this, we see that the 21st chromosome is very, very small, and so having an additional one here is not entirely catastrophic to the development of the individual. There's just a few abnormalities that are presented there in that condition as Down syndrome, but the individual can still survive and live a good life because of the fact that is a smaller chromosome. If trisomy or monosomy happened in either of these bigger chromosomes, it would likely result in a gamete that could not produce a viable offspring or a viable zygote. Okay, that's going to wrap it up here for meiosis.